Hey, it's Group 5. Let's talk about carbohydrates.
He knows a lot about the science stuff, Professor Dave explains.
We all heard of sugars and carbohydrates, especially when loading up energy for an exam or if we're on a diet. But on a chemical level, carbohydrates are made of smallest units called monosaccharides, such as glucose and fructose. Carbohydrates with two monosaccharides are then referred to as disaccharides. Today, we will look at the various experiments done to identify the properties and differentiate carbohydrates from one another based on their activity in each qualitative test.
As per the methodology of the qualitative test for carbohydrates, here are the list of materials that we need to incorporate in making this experiment happen. For our apparatus section, we need aspirator, aluminum foil or marbles or cotton to be used to cover the test tubes when heating them in the hot water bath to minimize evaporation, and beakers, Bunsen burner, hot water bath, iron ring and stand, matches or lighter, pasture pipette, test tube holder, test tube rack, and test tubes. Moving on with our reagents, we have 1 mole of fructose solution, 1 mole of glucose solution, 1 mole of sucrose solution. Barfield's reagent. This has an initial blue color and turns into a brick red precipitate once it detects a monosaccharide. Benedict's reagent. This also has an initial blue color that detects reducing sugars. When the sample turns green, it means that there are traces of reducing sugars, while orange in color means that there is a moderate amount of reducing sugars, and brick red precipitate when there is a large amount of reducing sugars. We will also be needing sulfuric acid, hydrochloric acid, distilled water, Felling's reagent. This is a deep colored blue solution that helps detect aldehydes, aldoses, and alpha hydroxyketones, such as fructose for example. And this eventually turns into a brick red precipitate when the sample is tested positive. Iodine solution. It has a reddish orange initial color but turns into a blue-black solution once a starch is detected from the sample. Molish reagent. This is used to identify and search for the presence of carbohydrates other than tetroses and trioses. It forms a purple ring at the interface that indicates the presence of carbohydrates. We will also be needing potato, which can be obtained from our household. Seliwanov's reagent. This reagent is used for detecting ketoses and has a cherry red color when a ketose is present in the sample. And lastly, unknown solution. In this experiment, we identified carbohydrates through different samples using a variety of qualitative tests. For the primary part of our procedure, we extracted starch from the potato. Firstly, the potato was peeled and grated. Afterwards, the potato gratings were washed in a bowl 3 times with approximately half a cup of distilled water. Distilled water was used to ensure accuracy and avoid impurities. After collecting the washings in a separate bowl, we allowed the mixture to stand for a few minutes until a layer of white sediment formed at the bottom. The excess liquid was discarded, keeping only 20 ml of the precipitate in a tightly locked container. This will now then be our potato starch solution. For the second part, we performed the hydrolysis for potato starch and sucrose. This is a chemical process that involves the breakdown of starch and sucrose into simpler sugar through the use of acid with the presence of heat and water. To execute this, we firstly dispense 4 ml of potato starch and sucrose into separate test tubes and labeled them accordingly. Second, we added 1 ml of concentrated hydrochloric acid in each of the test tubes and mixed well. Third, we covered the test tubes with cotton and heated them in the hot water bath for 30 minutes. This ensures that the molecules will disperse and act more thoroughly with the acid and water. This serves as our acid hydrolyzed starch and acid hydrolyzed sucrose. As for the part 3 of qualitative tests for carbohydrates, this section outlines various tests to identify and differentiate the different types of carbohydrates, such as monosaccharides, disaccharides, and polysaccharides. Each of these tests has specific outcomes and procedures. To start off, we prepared 5 ml of each sample, which are the unknown, glucose, fructose, potato starch, sucrose, hydrolyzed starch, and hydrolyzed sucrose, which are then placed in vials. For the Molish test, we dispensed 1 ml of each of our 7 samples and put them into clean dry test tubes separately. We then added 5 drops of Molish reagent to each while making sure that they are mixed well. Inside the fume hood, we carefully run 2 ml of concentrated sulfuric acid down the side of the test tube to prevent it from mixing with the sample and avoid interference with the expected results. A purple ring at the junction of 2 solutions indicates the presence of carbohydrates larger than tetroses. For the iodine test, we dispensed 1 ml of each sample and put it into clean, dry test tubes. We then added 1 ml of iodine solution. The resulting solution with blue-black color indicates the presence of starch. For the Barfield's test, we only dispensed 10 drops of each sample and again put them into clean, dry test tubes. We then added 1 ml of Barfield's reagent, making sure that they are mixed well. This time, we will be putting them into a hot water bath for 15 minutes. Heating the reaction mixture speeds up the hydrolysis of sugars. A presence of red precipitate indicates the presence of monosaccharide reducing sugars. For the Benedict's test, similar to the procedure from the previous test, we dispensed 10 drops of each sample in clean dry test tubes and added 1 ml of Benedict's reagent and mixed well. We then heated the test tubes in a hot water bath and observed the color they produced. Color transition from blue to red or orange indicates the presence of reducing sugars depending on the concentration. For Seliwanov's test, again, we dispensed 10 drops of each sample in clean dry test tubes, then added 1 ml of Seliwanov's reagent. We then heat them in a hot water bath. The yield of cherry red color indicates the presence of ketoses. As for our last test, the Felling's test, we dispensed 10 drops of each sample in clean dry test tubes and added 1 ml of Felling's solution, which was obtained from the mixture of Felling's A and Felling's B. Mixing these two solutions is an essential ground to create an active reagent that is effective for detecting reducing sugars. We then heated the test tubes in a hot water bath. The formation of brownish red colored precipitate indicates the presence of reducing sugars in the test solution.
As Rain mentioned earlier, the positive results vary depending on the qualitative test. Some detect ketoses, aldehydes, monosaccharides, reducing sugars, and even starch, with respective colors of the results. Additionally, it is important to note that some tests, such as Barfoed's, Benedict's, and Felling's tests, are closely similar to one another, but they still differ in some aspects. Felling's tests detect aldehydes, Barfoed's tests detect monosaccharides, while Benedict's test detects reducing sugar. After performing the qualitative test for carbohydrates, the changes in color were observed and the results were tabulated. All of the samples tested positive for Molish test, which is in line with the expected results since all of them were carbohydrates. For glucose, the sample tested positive for Molish test and Benedict's test. and yielded negative results for the rest of the test, indicating that the sample is a reducing sugar and the sample is also not a ketose. However, the sample should have tested positive in Barfoed's and Felling's test since glucose is a monosaccharide as well as an aldose. Moving on to fructose, the sample tested positive for Molish, Barfoed's, and Benedict's test, indicating that the sample is a reducing sugar and a monosaccharide. However, it should have tested positive for Felling's and Seliwanov's test since the sample is a ketose and a reducing sugar. Meanwhile, sucrose produced positive results for Benedict's and Barfoed's, but the sample should have produced negative as it is a non-reducing disaccharide. The difference between a reducing and a non-reducing sugars is that reducing sugars contain a free aldehyde or a ketone group, allowing them to transfer electrons to reduce other compounds, while non-reducing sugars cannot take part in reduction events due to the absence of free aldehyde or ketone group. Sucrose will not produce a positive result for Benedict's due to the lack of aldehyde and ketone groups because of the glycosidic bond connecting the anomeric carbons of glucose and fructose. For the past tests, the false result may have possibly arisen from cross-contamination or lack of the time for hot water bath, rendering the reagents ineffective to react with the test samples. On the other hand, the hydrolyzed sucrose produced positive for Benedict's and Barfoed's test, indicating that the sample is a monosaccharide and a reducing sugar. But the sample should have tested positive for Felling's and Seliwanov's test since there is supposed to be a presence of a reducing sugar from glucose and fructose. For the potato starch, the sample tested positive for the Molish and Benedict's test and negative for the rest. On the other hand, the hydrolyzed potato starch yielded positive results for iodine, Barfoed's, and Felling's test. However, it should have not produced positive results on the iodine test since the starch formerly present in the sample was converted into sugars due to hydrolysis. The false results in the previous tests may have arisen from the decrease in time of the water bath, resulting in partial hydrolysis, thus leaving some starch molecules. This explains the positive result for iodine test and negative for Benedict's test for hydrolyzed potato starch, while it is the other way around for unhydrolyzed potato starch. Lastly, the unknown sample only tested positive for Molish and Benedict's test, and negative for the rest. This indicates that the sample is a carbohydrate, the sample is a reducing sugar, it is not a starch, the sample is also not a monosaccharide, it is also not a ketose, and that it confirms that it is a reducing sugar such as an aldose. This concludes that the sample is a reducing disaccharide such as a maltose. In line with this, we can use the same approach to differentiate other carbohydrates. For instance, one can tell apart cassava starch from fructose through iodine and Seliwanov's test. For glucose and sucrose, we can make use of Benedict's test. The same goes for galactose and lactose, where we can use the Barfoed's test.
All things considered, qualitative tests help us to distinguish and identify carbohydrates from one another because of the structural variations between them. For instance, the iodine test distinguishes starch from the solution, Benedict's distinguishes reducing from non-reducing sugars, and the Barfoed's test distinguishes monosaccharides from disaccharides. These tests provide valuable insights into the composition of a sample and help determine the different classification of carbohydrates, such as monosaccharides, disaccharides, and polysaccharides, therefore helping us identify the presence and type of carbohydrates in the sample, which is indeed crucial for the fields of food science, medicine, and nutrition.
However, despite proper usage of laboratory equipment, following instructions, and wearing of protective suits, it is noteworthy to mention that the experiment was somehow still not 100% successful or fully accurate. It was apparent that the experimentation had some lapses, as some tests produced false positive or negative results. Hence, the group recommends to improve the following aspects. So the first one is to ensure that cross-contamination is avoided, where there are designated pipettes, beakers, and or dispensers for reagents and test samples. Therefore, avoiding the unhydrolyzed disaccharide sample test positive for monosaccharides. The second one is suggested to immediately subject the sample into hot water baths after adding reagent in qualitative tests that require heating. This is to activate the reagents, allowing the reaction to proceed efficiently. Without immediate heat, the reaction might not occur at all. And lastly, it is also suggested to invest more time for heating in some tests, since samples require more heat for hydrolysis to take place and the result to be more visible.